Hello everyone, my name is Akili Ligons and I'm a second-year PhD student in the Nidarian Immunity Lab, and today I'm going to be introducing coral cell transplantation, or more specifically, what questions need answering as we move forward with that. So every year, thousands of people receive life-saving transplants, whether it be a heart, a lung, a kidney, but before a surgeon makes their first cut, they must always ask themselves one key question: will this organ be accepted by the body? And when you think about it, rejection is the enemy of transplantation. So now in the context of corals, we see them exposed to this overwhelming heat stress, and cell transplantation comes in as this powerful tool to transfer desirable characteristics from a donor to a recipient. But there are a lot of questions that need answering before we can move forward with this. Do corals respond to each other at all? At what level do they respond to, whether it be the cellular or the tissue level or the whole organism level? And lastly, will this exposure induce an immune response? And that is where my research comes in. By observing the xenogeneic or cross-species cellular immune response, we're able to determine whether a rejection or an acceptance will occur. We measured this through a series of tissue dissociations, cell staining, and treatment exposure, followed up with fluorescent activated cell sorting, or FACS sorting. And this process enables us to not only detect but quantify these immune responses that we'll be observing. And specifically, we'll be looking at cytotoxicity, as shown on my right, your left, and phagocytosis. Cytotoxicity is programmed cell death, and phagocytosis is the engulfment, the active engulfment of perceived foreign material. And our results are quite promising. As you can see here, we're seeing no significant difference between the self-exposed or the xenogeneic exposed samples in cytotoxicity or phagocytosis. So what can we conclude from this? Well, a cellular transplantation appears feasible, but we're not getting the whole picture with just a cellular response, which is why my next steps are to incorporate the whole organism level response. And pending any consistencies or discrepancies, we'll be able to tell whether or not a transplant is truly feasible and achieve our ultimate goal of taking a susceptible recipient and making it a resistant one using resistant donor cells. Thank you.